This is Avenue with Vadin Kariyam Tahir. Today, the topic and the discussion in plant breeding and genetics is some basic key point about the agriculture biotechnology. So, let us start. Escherichia and agrobacterium are most useful in genetic engineering. Then, transposons genes are also called as jumping genes. Then uptake of plasmid and DNA. Plasmid DNA to bacteria is facilitated through CaCl, main calcium chloride. Next, gene expression and activation measured by marker gene. The next, for wall-free protoplasm, cellulase and pectinase required. Then cells proceed as follows: G1, S, G2 and then M. Next, DNA molecules identical except turns topoisomers. Then application of embryo application of embryo culture is overcoming of hybridization barrier. Then next, applied culture from pollen grain. Then differentiation of shoot in plant tissue culture is controlled by high cytokinin and oxygen ratios. Then next is about the genetic engineering and rDNA technology. Few of few key points as follows: DNA sequencing is by Maxam, Gilbert and Sanger DDSC method. Then vector have small size, multiple ori, low replication speed. Then for DNA staining, Fuelgen, Fuelgen, Feulgen is required for staining. EcoRI, Hind 3 and Bomb Hi1 are restriction endonuclease. Then alkaline phosphatase remove terminal phosphatase from 3 end. Then terminal remove terminal phosphatase from 5 end and then remove terminal phosphatase from both end. Then Taq polymerase in PCR is to use high thermostability. Then next, electroporation means rDNA in bacteria by Krant. Then next is Nick translation is done by DNA polymerase first, then telomeric sequence in YAC. Then next, plasmid isolation by alkaline lysis was published by Bimbin and Doly. Then Alec Jeffrey is related with DNA fingerprinting, and Kari Mullis is connected to PCR. Then group including those who made the first man-made rDNA molecules are included in Paulberg, Enmi, Chengi, Boyer and Stanley and Cohen. Then next is the 2006 Nobel Prize for RNA interference was given to C. elegans, elegans. Then rice is the first crop plant whose genome was sequenced. Then tobacco is the first transgenic plant. So now let us discuss about the PCR. Some key points as follows: Kari Mullis connected with the PCR, then DNA degradation, amplification, and sequences is connected with the PCR, billions of copies. Then machine is thermal cycler, then thermostable DNA polymerase required for PCR, then thermus aquaticus is source of Taq polymerase. Then next, thermostable polymerase are as follows: Taq polymerase, Wendt polymerase, and PFU polymerase. Wendt and PFU polymerase are more efficient than Taq polymerase because of proofreading activity. Then next, requirement of PCR are DNA segment to be amplified. Next, 2 oligonucleotide primers. Then next, 1 heat stable polymerase, and there are 3 steps in PCR are as follows. First step, denaturation at 90 to 98 centigrade for heating. Then second is annealing, binding primers to denatured stand at 40 to 60 centigrade. Then next, third point is primer extension or elongation at 72 centigrade temperature. Then asymmetric PCR used to germinate single strand copy of DNA sequencing, then used in site directed mutagenesis, then remove transcriptase PCR, mRNA as template to cDNA, from cDNA then use amplifying RAPD map detect transgenes. So this was all about some key points about PCR. Thank you.